my name is Keandra Kane, and this is my presentation on dopamine metabolism and its dysregulation in Parkinson's disease. This presentation focuses on how dopamine metabolism works normally in the brain and how it becomes disrupted in Parkinson's disease. Dopamine metabolism includes every step from synthesis to storage, release, degradation, and reuptake. In Parkinson's disease, problems in these metabolic pathways, such as impaired synthesis, excessive breakdown, reduced vesicular packaging, and increased oxidative stress, contribute to the death of dopamine-producing neurons. Understanding these mechanisms is crucial because it helps explain both the symptoms of Parkinson's disease and why dopamine neurons are especially vulnerable. Parkinson's disease is a neurodegenerative disorder that primarily affects motor control. It's most common in older adults, with about 1% of individuals over 60 affected. The disease is characterized by the loss of dopaminergic neurons in the substantia nigra pars compacta, which supply dopamine to the striatum. When these neurons die, dopamine levels drop sharply. This dopamine deficiency disrupts normal signaling in motor circuits, leading to tremors, rigidity, and slow movements. There are also significant non-motor symptoms such as mood changes, sleep disturbances, and cognitive decline, showing that dopamine affects many brain areas beyond movement. The nigrostriatal pathway is the major dopamine pathway responsible for voluntary movement control. Neurons in the substantia nigra send long projections to the striatum. When these neurons fire, dopamine is released into the striatum and modulates the activity of key motor circuits. In Parkinson's disease, these projections degenerate, which means the striatum receives less and less dopamine. This breakdown directly correlates with the motor symptoms of the disease. Even before cells fully die, early metabolic defects weaken dopamine release and synaptic function in this pathway. Dopamine synthesis begins with the amino acid tyrosine. Tyrosine hydroxylase, or TH, is the enzyme that converts tyrosine into L-dopa, which is the rate-limiting step. This means TH controls how much dopamine can be made. From L-dopa, AADC converts it into dopamine. In Parkinson's disease, TH levels or activity often decline due to stress on the neurons, including oxidative stress and problems with gene regulation. So one metabolic issue in PD is simply that dopamine cannot be synthesized as efficiently as before. Once dopamine is made, it has to be stored safely. Dopamine is actually chemically unstable in the cytosol and can form toxic byproducts if not properly packaged. VMAT2 is the transporter that pumps dopamine into synaptic vesicles where it's isolated and protected. In Parkinson's disease, VMAT2 levels or function may decrease. When VMAT2 doesn't work properly, dopamine accumulates in the cytosol where it oxidizes and produces harmful molecules. This oxidative stress damages the neuron and contributes to its eventual death. Dopamine breakdown is primarily handled by the enzyme MAOB, which converts dopamine into DOPAC while generating hydrogen peroxide. Hydrogen peroxide is a reactive oxygen species that can damage cell structures. COMT participates in further degradation. In Parkinson's disease, MAOB activity can increase, which means more dopamine is broken down and more oxidative stress is produced. This adds to the burden on dopamine neurons and creates a cycle where breakdown products worsens the health of the cell. After dopamine is released, the dopamine transporter DAT clears it from the synaptic cleft. This recycler is important to maintain precise control over dopamine signaling. DAT also helps conserve dopamine by reusing it. In Parkinson's disease, DAT function becomes altered, affecting how much dopamine is available for signaling. Another key regulator is DJ1, a protein that protects neurons from oxidative stress. When DJ1 is mutated or dysfunctional, as seen in some forms of PD, neurons cannot handle the oxidative byproducts of dopamine metabolism, making them more likely to degenerate. Dopamine dysregulation in PD is multifactorial. First, fewer dopamine neurons means less dopamine can be produced or released. Second, there's also evidence that MAOB becomes overactive, increasing metabolic stress. Third, reduced VMAT2 functions raise cytosolic dopamine, which leads to toxic oxidation. Fourth, mitochondrial damage further reduces the cell's ability to handle oxidative stress. All these factors create a harmful cycle in which dopamine metabolism itself becomes toxic, leading to even more neuron loss. Some forms of Parkinson's disease are linked to mutations in genes such as DJ1, Parkin, and PINK1. DJ1 is especially important because it acts as an antioxidant and helps maintain mitochondrial function. When DJ1 is mutated, the cell becomes more sensitive to dopamine-induced oxidative stress. Similarly, other PD-related genes disrupt mitochondrial quality control or impair dopamine handling. Environmental toxins like MPTP can mimic the effects of these mutations by directly damaging the mitochondria and disturbing dopamine metabolism. These genetics and environmental factors show how tightly dopamine metabolism is linked to neuron survival. When dopamine isn't safely stored, it oxidizes into dopamine quinones and other reactive species. These molecules can bind to and damage proteins, lipids, and DNA. They also directly impair mitochondria, reducing ATP production and pushing cells towards death. Dopamine oxidation can also promote the misfolding and aggregation of alpha-synuclein, which forms Lewy bodies, a hallmark of PD pathology. Importantly, symptoms do not appear until dopamine levels have dropped about 60 to 70%, meaning much of this metabolic damage occurs silently before diagnosis. In the basal ganglia, dopamine normally boosts the direct pathway, which facilitates movement, and suppresses the indirect pathway, which inhibits movement. When dopamine levels falls, the direct pathway weakens and the indirect pathway becomes overactive. This creates excessive inhibition of the thalamus, making it difficult to initiate movement. Imaging studies have shown reduced activity in dopamine-dependent circuits in people with PD. Some compensatory receptor changes occurs, including increased D2 receptor sensitivity, but they are not enough to restore the normal movement. The most widely used treatment for Parkinson's disease is L-DOPA, which bypasses the damaged dopamine synthesis pathway. MAO-B inhibitors slow dopamine breakdown, reducing oxidative stress and extending dopamine's availability. COMT inhibitors helps L-DOPA stay active longer by preventing peripheral breakdown. Dopamine agonists mimic dopamine at its receptors. These treatments address the consequences of dopamine loss but do not stop neuron degeneration. They also come with side effects, including dyskinesias with long-term L-DOPA use. Researchers are exploring therapies that target earlier stages of dopamine metabolism. Antioxidants like T polyphenols can reduce oxidative stress and protect dopamine neurons. Gene therapy aims to restore enzymes involved in dopamine synthesis or proteins like DJ1 that help cells manage stress. Other strategies focus on increasing VMAT2 to reduce cytosolic dopamine toxicity. Additionally, treatments that stabilize mitochondria or prevent alpha-synuclein aggregation could slow or prevent the metabolic cascade that leads to neuronal death. These approaches aim to go beyond symptom management and actually modify disease progression. When we step back and look at everything discussed so far, it becomes clear that dopamine metabolism sits at the very center of Parkinson's disease pathology. Normally, dopamine is synthesized, packaged, released, and broken down in a tightly controlled cycle that supports healthy motor circuit function. But in Parkinson's disease, this balance collapses. Dopamine levels fall dramatically because the neurons responsible for producing it, those in the substantia nigra, are progressively lost. The downstream effects are far-reaching. Oxidative stress increases, mitochondria becomes damaged, toxic dopamine metabolites accumulate, and alpha-synuclein aggregation accelerates neurodegeneration. What we now understand from recent research is that these metabolic and oxidative pathways are not byproducts of Parkinson's disease. They are active drivers of disease progression. This is why so many therapeutic strategies, both current and emerging, focus not just on replacing dopamine, but on protecting dopamine handling systems. limiting oxidative damage, stabilizing mitochondria, and also strengthening the cell's natural defenses. A deeper understanding of dopamine metabolism, how dopamine is synthesized, handled, and broken down, will allow researchers to identify new therapeutic targets. This includes interventions that boost VMAT2 to safely store dopamine, drugs that reduce MAOB associated oxidative stress, antioxidants like T polyphenols that neutralize ROS, and gene therapies that restore protective proteins like DJ1. Each of these strategies reflect the same principle. If we can preserve dopamine metabolism and protect the neurons that control it, we can potentially slow or alter the course of Parkinson's rather than just manage the symptoms. In summary, we explored how Parkinson's disease begins with the degeneration of dopaminergic neurons and the substantia nigra, and how dopamine depletion disrupts the basal ganglia motor circuit. We walked through the dopamine synthesis, storage, degradation, and regulation, and we saw how dysfunction in any of these steps contributes to a harmful cycle of oxidative stress and neuronal vulnerability. We also discussed the molecular contributors like DJ1 and the pathological effects of dopamine oxidation, mitochondrial damage, and alpha-synuclein accumulation. Finally, we examined current treatments that aim to restore dopamine levels and emerging therapeutic approaches targeting the underlying metabolic and oxidative causes. Altogether, understanding these mechanisms does more than explain the symptoms of Parkinson's disease. It points towards the futures of more effective and potentially disease-modifying therapies.